Hello, fellow neuroscience enthusiasts. This video is another installment of the Brain Preservation Foundation's Neuroscience Journal Club, in which we briefly review the results of a selected paper and discuss its relevance to the ongoing debate regarding brain preservation. Today's paper is entitled Spatial Connectivity Matches Direction Selectivity in Visual Cortex by Rossi, Harris, and Carandini. It was published in the journal Nature on November 11, 2020. A link to the paper is listed below. This is a really fantastic paper that I highly recommend reading. It addresses a question that has obsessed neuroscientists since at least the classic circuit models proposed by Hubel and Wiesel in the early 1960s. Specifically, are the receptive field properties of visual cortex neurons determined by their pattern of synaptic connectivity? And if so, in what way? The Rossi paper brilliantly addresses this question as directly as I have ever seen by recording the receptive field properties of a single layer 2-3 cell in mouse visual cortex and simultaneously recording the receptive field properties of all of the layer 4 and layer 2-3 cells that are presynaptic to this target cell. They achieved this amazing feat using an incredibly powerful viral tracing technology invented separately by Wickersham et al. Unfortunately, I will only be able to cover a fraction of what is in this paper, so please do try to read the full paper yourself. Also, please note that although I will try my best to accurately relate the research, it is always possible that I may misinterpret something crucial. If I do, please let me know in the comments, and I will try to correct it. Layer 2-3 visual cortical neurons are typically selective to visual stimuli, like edges, which have a specific orientation and are moving in a particular direction. It has long been proposed that such selectivity is determined by synaptic connectivity, as Hubel and Wiesel did in their classic paper from 1962. They suggested that the orientation selectivity of layer 4, quote, simple cells is determined by their synaptic pooling from laterogeniculate nucleus projections with co-aligned receptive fields. And they proposed that layer 2-3, quote, complex cell responses are determined by their synaptic pooling of layer 4 projections with co-aligned but offset receptive fields. A wide range of experiments over the years have supported this hypothesis, but key questions remain. Rossi et al. Briefly review the state of the field at the start of their paper. Reading from their introduction. The selectivity of layer 2-3 neurons of the primary visual cortex, V1, for orientation and direction has unclear circuit origins. Selectivity is thought to emerge from the pooling of co-tuned layer 4 excitatory neurons, amplified by excitatory connections between co-tuned layer 2-3 neurons. However, it remains unclear whether the inputs from layer 4 are tuned to the appropriate orientation, and whether they dominate or complement inputs from layer 2-3. Orientation tuning might be sharpened by inhibition, but the role of inhibition is controversial, and inhibitory connections can be promiscuous and unspecific. Moreover, none of these mechanisms explain direction selectivity, because the direction preference of layer 2-3 neurons often differ from their intercortical inputs. Direction selectivity might arise from tuned inhibition or from the spatial offset of excitation and inhibition, but these circuits have not yet been demonstrated. To summarize, these are the main questions that Rossi et al. Will answer. 1. Is the orientation selectivity of a layer 2-3 cell sufficiently predicted by the excitatory layer 4 cells presynaptic to it? And 2. How is the direction selectivity of a layer 2-3 cell determined? The authors address this question by injecting a specially designed rabies virus into a single layer 2-3 cell in a mouse, which then infects all the cells and only the cells that are presynaptic to that target cell. All the cells express the green calcium indicator GCAMP6, which allows all of their receptive field properties to be recorded. And doing so allows them to test the hypothesis that the target cell's receptive field is formed by simply pooling the receptive fields of the cells presynaptic to it, or if something more complicated is happening. The viral tracing method the authors used was based on one developed in this landmark paper by Wickersham et al., 2007. Here's how that paper describes the technique. Here we present a transsynaptic tracer that crosses only one synaptic step, unambiguously identifying cells directly presynaptic to the starting population, in this case a single cell. The core idea of this system is to infect the cell of interest with a deletion mutant tracing virus that is missing one or more genes that are required for transsynaptic spread, and to complement this deletion by providing the missing viral genes in trans in the initial infected neurons. With all the viral genes present in the starting cells, the virus can spread from them to cells in monosynaptic contact. Because those genes are not in the secondarily infected cells, however, the virus cannot spread beyond them. A normal rabies virus will spread exclusively in the retrograde direction, crossing over to all presynaptic cells that synapse onto the infected cell. However, with a normal rabies virus, the spread will continue from these cells as well, labeling cells separated by 2, 3, or more synaptic jumps from the target cell. Since the rate of spread is variable, it is not possible then to distinguish between the cells directly connected to the target cell by a single synapse from those connected by multiple synaptic jumps. Wickersham et al. Created a modified rabies virus by deleting the rabies virus glycoprotein gene. The glycoprotein is not required for replication, but it is required for transsynaptic spread of the virus. They replaced that gene with one that coded for green fluorescent protein so that they could see which cells the virus replicated in. Wickersham et al. Also used a rabies virus that would only infect cells that expressed the TVA viral receptor, which is a protein which is found only in birds and not in mammals. Here's a diagram based on that Wickersham paper, showing a target cell surrounded by other cells that are either directly synapsing onto the target cell or removed from it by two synapses. The goal is to label only the ones immediately synapsing onto the target cell and no others. First, two genes are inserted into the single target cell via electroporation. One of these genes is for the TVA receptor, and the other is for the rabies glycoprotein. After a short time, this causes TVA receptors and rabies glycoproteins to be expressed in the target cell only. Now, the modified rabies virus is injected so that it spreads across all of the cells, but it can only infect our one target cell that is expressing the TVA receptors. After a short time, the modified rabies virus replicates within our target cell. And because this target cell also contains the rabies virus glycoprotein, these viruses are able to infect cells directly presynaptic to our target cell. But this spread goes no further because the newly infected cells do not have the rabies glycoprotein needed to pass across their synaptic partners. Because all of the viruses contain a green fluorescent protein gene, they will all show up as brightly labeled. Now back to our Rossi et al. Paper. They started with a mouse that expressed the calcium indicator GCAMP6 in all cells so that the visual responses could be recorded using two-photon imaging. They electroporated a single target cell with the TVA and rabies glycoprotein genes, but included the gene for the fluorescent marker DS-Red as well. Then they used the same modified rabies virus trick to label all presynaptic cells to this target cell. This figure from the Rossi paper shows the target cell in yellow and all presynaptic cells in red, with other cells shown in green. Rossi et al. Used a few other tricks to identify cells as being either excitatory or inhibitory, but you'll have to read the paper to get those details. They showed the mouse images of moving gratings with different orientations so that they could plot the receptive fields of the target cell, as shown here, all of the excitatory cells presynaptic to that target cell, as shown here, and all of the inhibitory cells presynaptic to that target cell, as shown here. Figure B here shows target cell receptive field responses from two different mice. Let's make sure that we understand these plots. 0 degrees is defined as this particular cell's preferred grading movement direction. So all direction selective cells are going to have a peak response at 0 degrees by definition. And the x-axis angles are relative to this preferred direction. 180 degrees is the same grading orientation, but moving in the opposite direction. So the difference between the response at 0 degrees and at 180 degrees is actually a measure of the direction selectivity of the cell. They also separated cells based on whether they were in layer 4 or layer 2-3. Figure E here shows the combined tuning of all excitatory presynaptic cells. Note that this is the same type of tuning plot, but pooled over all presynaptic excitatory cells. Figure G shows these responses broken into whether they came from layer 2-3 cells or layer 4 cells. And figure J here shows the combined tuning of all the inhibitory presynaptic cells. Rossi et al. Imaged 16 mice in total. And figure C here shows the average of all the target cell responses, of course with all of their preferred directions aligned at 0 degrees before this averaging. As expected, the layer 2-3 cells show a robust orientation and direction selectivity. Figure F here shows the average of all the excitatory presynaptic ensembles. Now this is a key figure. It shows that the presynaptic cells' orientation responses are significantly predictive of the target cell's response, implying that there is a causal role in the target cell's response. But the presynaptic cell's direction selectivity is not significantly predictive. Figure H shows the same is true when considering layer 2-3, and layer 4 cells separately. And figure K here shows that the inhibitory cell responses are not significantly predictive of either orientation or direction selectivity. The key result here is that the orientation tuning of our target layer 2-3 cell can be explained by the synaptic connections it makes with a subset of layer 4 cells. This verifies the prediction made by Hubel and Wiesel in 1962 that we discussed earlier. But the direction selectivity of that layer 2-3 cell cannot be explained in such a manner. Rossi et al. Go on to address this question of where layer 2-3 direction selectivity might come from. Here is a plot of one target layer 2-3 neuron showing its direction tuning using a polar plot. Here is a plot showing all of the excitatory in red and inhibitory in blue presynaptic neurons of that target cell, which is itself depicted as an open arrow in the center here. This plot shows where those cells are relative to each other in the cortex. The scale bar is 200 microns here. The authors painstakingly then determined the retinotopic positions of each of these cells, that is, the position in visual space that they were tuned to. And this plot shows all of the excitatory presynaptic neurons plotted in retinotopic space relative to the target cell's retinotopic position. The scale bar here represents 5 degrees of visual angle. And this plot does the same with all the inhibitory presynaptic neurons. Now, if you look closely, you can see that the excitatory cell's receptive field locations are shifted slightly ahead of the target cell's position. That is, a bar moving in the preferred direction for this cell will excite these presynaptic cells slightly before they do our target cell. And the inhibitory cell's receptive field locations are shifted slightly behind the target cell's location. That is, a bar moving in the preferred direction will excite these inhibitory presynaptic cells slightly after they do our target cell. Rossi et al. Made similar plots for all 16 mice imaged in this way, and aligned all of the plots in visual space so that the preferred direction of the target cells was to the right. Now it is easier to see that a bar moving to the right will generate an excitatory presynaptic response a bit earlier, followed by a later inhibitory presynaptic response. The authors quantify this offset here. They also show that the receptive field locations of the excitatory cells are elongated parallel to the preferred direction of the target cell. Other research has shown that the inhibitory currents rise in layer 2-3 neurons later than the excitatory currents. And this differential delay of about 10 milliseconds, along with the offset of receptive fields, provides an explanation of the direction selectivity seen. If the bar is moving in the preferred direction, then the excitation is not canceled by the inhibition. But if the bar is moving in the opposite direction, then the excitation is canceled by this inhibition that's delayed, so the target cell does not respond. For each of the 16 cells, this excitatory to inhibitory offset was measured. And this was used to predict the strength of the direction selectivity. This plot shows that the predicted strength of direction selectivity, predicted based upon the connectivity, was well correlated with the actual measured strength of direction selectivity of those cells. Rossi et al. Summarized their findings like this. We have revealed a precise spatial patterning of excitatory and inhibitory connections to layer 2-3 neurons, which can strengthen orientation tuning and establish direction selectivity without connections from co-tuned neurons. The standard model for selectivity in layer 2-3 relies on excitatory co-tuning. However, we have shown that the direction preference of a layer 2-3 neuron does not correlate with that of its presynaptic excitatory cells. Instead, it strongly correlates with the spatial pattern and offset of excitatory and inhibitory inputs. Given the delay between excitation and inhibition, this offset may explain direction selectivity in layer 2-3. I hope you agree with me that this is a truly fantastic paper. It demonstrates how a powerful genetic tool, the modified rabies virus technique, can be used to address fundamental questions about how the brain works to process visual images. Now, let me very briefly address what I see as the relevance of this particular paper to the ongoing brain preservation debate. A concern I often hear regarding brain preservation is you cannot determine the functioning of the brain from only a static map of its connectivity. Now, of course, no single neuroscience result can adequately address such an open-ended objection. Only a thorough review of the last perhaps 50 years of research could attempt to do that. The whole collection of these Journal Club videos is designed to kind of take a stab at such a review. Nonetheless, I think the Rossi et al. Paper just discussed does address this question, at least in a small way. It showed that the receptive field properties of a visual neuron can, at least in part, be predicted based upon connectivity alone. And it stands as part of a growing number of research papers that have shown similar results. And we should remember that they did not have anywhere close to the true connectivity. The Rossi et al. Paper had no information on the number of synapses each of the presynaptic neurons provided. They had no information on the sizes of those synapses, along with any other ultrastructural features of synapses that we know predict functional strength. They had no information about where each synapse was located on the target cell's dendritic arbor, or the cable diameters of the arbor that might influence that synapse's effect on the target cell's activity. We should note that all of those classes of information will be available in, for example, EM Connectomics plus functional imaging studies of mouse V1 that are currently being run at places like the Allen Institute. And the Rossi et al. Paper certainly did not have any information on the exact ion channel and receptor distributions on each synapse or each part of the target neuron's arbor. In contrast, a brain preservation technique like aldehyde-stabilized cryopreservation seems capable of preserving all of these structural and molecular features. So it could be expected that a more accurate prediction of function should be possible to achieve, at least in principle. However, a counter-objection to consider is that the Rossi et al. Predictions of the layer 2-3 cell's receptive field properties were based on knowing the true receptive field properties of the cells just presynaptic to it, something that would not be known in a brain preservation scenario. Let's consider for a moment what it would really take to, of course in the far future, simulate the ventral visual stream of a preserved brain. The first step would be to assume a set of receptive field properties in some early visual areas based on anatomy alone. For example, if the retina was preserved, then it would be straightforward to determine the receptive field properties of all the retinal ganglion cells based on their locations, morphologies, and of course, connectivity. From there, LGN receptive field properties could be predicted. Then primary visual cortex V1 properties could be predicted, and likewise for later areas. A simulation based on this would, of course, model not only the feed-forward connectivity, but also the feedback connectivity, along with all relevant modulators, etc. A key question in such an approach is how parameter uncertainties will accumulate. Will they compound so that the receptive field properties of later visual areas will essentially bear no resemblance to the original biological ones? Or will the nervous system's built-in redundancy and attractor dynamics ensure that minor inaccuracies in the assumed parameters do not build up catastrophically, similar to how other sources of noise are known to be suppressed by network dynamics? We simply don't have enough information to answer these questions as of yet, but they could be addressed through existing research methods, like the ones in the paper we discussed here. And they could be addressed via new computational modeling. For example, by injecting specific amounts of noise into the parameters of deep recurrent neural network models of the visual system. In this way, researchers could learn just how precise these parameters need to be known. For example, if the excitability threshold of every neuron in such a model was changed by a few millivolts, and in such a model, would a picture of a cat still be recognized as a cat? What if the functional strength of every synapse was changed by, say, 10%? Such studies are crucial if we are to have a science of brain preservation. They are needed to understand what structural and molecular features must be preserved and with what fidelity. And they are needed to understand how advanced the future imaging technologies must be in order to allow accurate simulation of the preserved brain. Thank you for listening, and thank you for keeping an open mind regarding what advances in neuroscience might bring in the future. These videos are designed to spark conversation and to spark debate, so please feel free to leave comments below. I look forward to reading them.